Hello. In the following video, we will do the question on monoclonal antibodies. So by the end of this series of videos, you will be very good at this monoclonal antibodies. So let's see what we have. Now I want to explain this, whatever happens over here, so that you get this direct, you don't be in blue when we talk about these monoclonal antibodies. Okay, so let me just bring for you a picture. So yeah, here we have a mouse. Okay, this mouse, we take the plasma cells from the spleen of the mouse. First thing we do is we inject. I guess you know that we're talking about production of antibodies. Okay, so with image you will be able to understand more clearly. So the first thing is antigen is injected. Remember, mind you, that this is the antigen which we are which we are trying to make the antibody of. Okay, so antigen is injected, and the mouse B lymphocytes that recognize the antigen proliferate and form plasma cells. Okay, they form plasma cells in the spleen. Okay, we take this plasma cells from the mouse, and then we take cancer cells. We join them, we get a hybridoma cells. Okay, and then tiny samples are taken so that there is only one cell present in a well. Every well is tested so that any hybridoma cell that gives the required antibody can be found. Okay? A clone of cells is produced which only makes this one antibody. So this is actually very simple. We take we inject, for example, we have an antigen. Okay, we have an antigen. This one is an antigen. Okay, okay, wait, let me just keep it this way. This one have a receiver. This one, it has a receiver. This one. Okay, this one is an antigen. So we will take this, we will keep it inside the mouse. Okay, we will inject this inside the mouse. So the mouse spleen over here, the spleen will produce the antibody for this antigen. So the antibody for this, which is the plasma cells, the antibody will be like this, right? It will be the complementary of this. Okay, it can get attached over here. Sorry, this one won't get attached. We need something real. So the thing that will get attached is this one. Okay, okay, so this one can get attached over here. So so this one then is we inject. This one is in the spleen. Okay, we take this out, and we bring this to the this one. Okay, plasma cells in the spleen, and we take another new cells from the cancer cells, and then we join this and we get a hybridoma cell. Okay, after getting the hybridoma cell, we move this and then we clone the cell to produce many antibodies. So this point is clear. Now let's see what does this tells us. The step 1, it tells us mouse myeloma tumor cells are cultured in a medium that does not contain hypoxanthine aminopterin and thymidine. Okay, mouse myeloma tumor cells. So this is a hybridoma cell. Okay, a mouse is injected with an antigen. Spleen cells. These are tumor cells. It already says here. This is a tumor cells. And then a mouse is injected with an antigen. Spleen. This is the process we said over here. It's injecting. Okay. So mouse is injected with an antigen, and then spleen cells are removed from the mouse and cultured. These cells will survive in a medium containing HAT, right? So spleen cells are removed from the mouse and cultured. So we take out the spleen cells and we culture it, and these cancer cells. So these are cancer cells, Okay, these are cancer cells, and this is a plasma cell. Okay, plasma cell. We get a mutant myeloma, and spleen cells are fused. Okay, we get a hybridoma. The fused cells are grown into a culture medium. This is the cloning, we said. Okay, this is the cloning. This is the hybridoma. Hybridoma, right? And then the surviving cells are separated and cloned to give different cell lines, each secreting a particular monoclonal antibody. Okay, the clones are tested for binding of the antibodies with the antigen, and the most effective selected for production. So name this method of producing monoclonal antibodies. I think it's the hybridoma method, A1. Hybridoma method, right? So we have to mention this as a, and that's why they didn't mention the term hybridoma here. So this is a hybridoma method. Hybridoma. Make sure you know the spellings well, okay? And then describe what is meant by the term monoclonal antibody. So for this case, monoclonal antibody, what is it? Monoclonal antibody is identical or produced by cloning, and variable regions, antigen binding site, all identical for antigen. So when we talk about monoclonal antibodies, is the one that is identical to the antibody which would be produced. Okay, it has the same binding sites. So monoclonal antibodies has the same binding sites. So we can talk about binding sites. Okay, and then we can also talk about the antigen binding sites. Okay, identical antibodies produced by cloning. Okay, the same binding site or same variable, same we have antigen binding site, same antigen binding site, antigen binding site, antigen binding site same, and then also we have this variable regions, antibody specific to one, variable regions. In short, these are the characteristics, but the main thing to mention when we are told to define monoclonal antibodies are that these are the ones that are identical completely. So because why are they identical? Because they are produced by cloning. So like if you have your one antibody A, okay, and you have your B, okay, this won't be identical, but here we just take this A and then we produce everything from this A. We don't meet it with anything else. Okay, so eventually what you will get is a monoclonal antibody, the one with one type of, like a single parent, in other words you can say. So then it just simply means that it is a single, all are identical antibodies. Okay, so the definition would go identical antibodies, okay, produced by cloning, having the same binding sites. Antibodies produced by cloning. Okay, so you can talk about this point, then you can go to this point, and then lastly to this point. Okay, hope I made myself clear there. And then they give you, describe in detail the molecular structure of one of the antibodies produced in step 6. You may wish to use an annotated diagram to answer the question. Now here, if you remember, in AS you must have drawn the antibody. It goes like something like this, right? I just decided to get you a diagram from the net. So here we have the diagram. We have antigen binding sites, we have variable region, and then we have light chains and heavy chains. So you should draw like this. If you want to get to know how to draw, you can start. You can use a black ink. Okay, let me use a black ink. Okay, you can start with this region first, like this, and then you can go on this way. Okay, and then again you can come all the way here, and then you can go on this way. So that's not good. Maybe I can just copy this and copy this and then paste this. Paint is really fun to use actually. Okay. That's not the one which I wanted. I wanted to flip horizontal, yeah. So we had the same exact structure, you see. Then we can have another structure over here, something like this. Okay, something like this, right? Then we can have the antigen binding sites over here. So this region over here, this region is where the antigen, this is the antigen binding site, right? This is the antigen binding site, and we have heavy chains, and we have a variable region here. This one always change. We have a variable region, and lastly we have this light and heavy chains. Okay, we have these ones, heavy, and we have these ones, light. Okay, light chains. Yeah, so hope you understood the structure. So with that, having said that, I will continue the number C in the next video. Thank you for listening.